Molecules that are too large or polar cannot diffuse across the membrane, and thus they need some type of assistance into penetrating that membrane and entering the cell. This includes amino acids, glucose, and other organic molecules, where we'll need some type of carrier-mediated transport. And this is done by the membrane carrier flipping its shape or conformation. And so this can be active or passive, and when we say active, this means that it would require energy versus passive that does not require energy. There are certain carrier proteins that we find within the plasma membrane that move these molecules across. And so characteristics then of these include that they are specific to a certain molecule. There may be competition for similar carriers or molecules, and there might be saturation where the number of carriers is limited no matter how many molecules are present. So the types that we have for assisted membrane transport include facilitated diffusion, active diffusion, and then vesicular transport. So with carrier-mediated transport, the rate of transport of the molecule into the cell is directly proportional to the extracellular concentration of the molecule. And so that will occur, so in this graph here we have concentration of X and then the rate of transport on the y-axis. So this will occur until we hit saturation. At this time, the rate of the transport reaches a maximum value. So this is at max. And so the rate of the transport does not increase with an increase in the extracellular fluid concentration of the molecule. So you can keep adding this molecule, but it won't matter. And so here's another example of, say, this ion has a saturation rate that's higher than that of, say, this molecule. So that's just what this figure is showing. Another example is facilitated diffusion, and this is where substances move from a higher concentration to a lower concentration. However, this requires some type of carrier molecule. And so here within the plasma membrane, we have this carrier protein. And this is the means by which glucose is transported into cells. And the transport proteins may always exist in the plasma membrane, or they might be inserted when needed. And so this, though, requires no ATP, so it's not considered to be active. Rather, this is going to be a passive type of diffusion, where we have, again, outside of the cell, higher concentrations, then inside of the cell at lower concentrations here. Okay. All right, a third type we have is active transport, and so this is where we need energy. And sometimes molecules must be moved from an area of low concentration to an area of high concentration. So this means that they move against their concentration gradient. So you're going from low concentration to high concentration. And so this too will require some type of carrier molecule. Sometimes these will be occurring through different types of pumps. Hydrogen pumps are examples of this, and sodium-potassium ATPase pumps are also an example of this as well. And there are two types of active transport. So this first one, the primary active transport, so this will use ATP directly, and so this will transport protein is also the enzyme ATPase that is used, and it will hydrolyze ATP. The second type we have is the secondary active transport, and this is driven by an ion concentration gradient that's established by the primary active transport system. A great example of this is a sodium-potassium ATPase pump, which we'll see within neurons, but also within in the cardiac cells and in several other systems. And so this is found in all body cells, but serve a very important role, especially within the nervous system. And so the ATPase enzyme, what it does is it pumps 3 sodium out of the cell and then 2 potassium back into the cell. And this is against their concentration gradients because potassium is highly concentrated inside the cell as compared to outside of the cell, and then sodium is actually in higher concentrations outside of the cell versus inside the cell. And so when we have this enzyme, ATPase, what it does is it hydrolyzes ATP to ADP and then blocks both openings. And so when the ADP is released, what we have then is a conformational shape change that allows 3 sodium to move out of the cell and then 2 potassium into the cell. And so the pump returns to original shape once this has completed. And the important thing is, so for the neuromuscular system, this helps to create electrochemical impulses, but then also within other cells of the body helps to maintain osmolality. With the secondary active transport, there's also another type that is known as the coupled transport. And this is where we have, say, 2 molecules that need to bind in order for transport of material to occur. And so this is true where since it's active, we're moving molecules against their concentration gradient. And so this is considered to be a co-transport or symport, where one molecule, for example, moves with sodium. And so glucose, what we have here, has to move and bind with sodium into this carrier protein before it can enter into the cell. Okay, and we see this a lot with digestion of materials actually, where having glucose enter from the bloodstream into the cell, we need to have sodium as well for that to occur.